Hello, lovelies, back in your classroom. We left off with osmosis, which remember is the diffusion of water in or out of something that has a soluble permeable membrane. You have to have water, you have to have a semi-permeable membrane. Those are the two things going on with osmosis. And this is really what's happening in plants. You have water going up and down in the xylem, and you have the xylem and phloem, which have semi-permeable membranes. Let's talk about water potential. So water potential is a measurement that actually combines solute concentration and pressure. So how much pressure is acting on the water? What's the pressure of gravity? What's the pressure of transpiration? What's the pressure? How small is that straw? So a lot of things happen there. There's an equation, there's all kinds of fun things. You don't have to do any of that. Settle down. All I want you to know is that in a plant, because it's a closed system, there's pressure. There's actual pressure on the water. This is pressure, it wants into something and it can't get in there because it's not semi-permeable. There's pressure by the roots, there's pressure from transpiration, there's actually pressure. And this is the water potential. This is the amount of energy that the water has to do work, to actually do things in the cell. So don't panic about all of the equations, just know that water potential determines which way the water is going to move. So this is the sentence that we're looking at. Water potential, underline this guy. This is what we need to know. Okay, it's abbreviated in megapascals. Don't panic, we're not doing the equation. Settle down. Dude, just know this little guy here is a megapascal, and it's what we measure water potential in. That's it. Chill out. I don't need you to know anything else. Okay. Let's move on to the protoplast. So you can skip all the other parts in your notes. So there should be some parts just about the equation. Don't worry about that equation. Just worry about megapascals. Let's move on to the protoplast, shall we? The protoplast. This is the living part of your plant cell. Here's your plant cell. This is the living part of the cell, which includes the plasma membrane. There's your plasma membrane. Here's your plasma membrane. The protoplast is this outside part, right? It's the living part, which includes the plasma membrane. So that's what we're looking for. Let's go on to the next one. Turgor pressure is the pressure exerted by the plasma membrane against the cell wall. So when we talk about the protoplast, we're going to talk about sort of this part in here, if you will, this outside part. And this is going to be your plasma membrane. So what you're going to have is you're going to push against this and you're going to get a turgid cell. So you're going to get a big fat happy cell that's stretched out as much as it can be. It's all fluidy and happy. This is turgor pressure. This is the amount of pressure being pushed up against the cell because it's all full of water and good stuff like it's supposed to be. So this is the pressure exerted by the plasma membrane against the cell wall and the cell wall against the protoplast. So all these things are pushing, as the water fills it, it's pushing out. Water potential affects the uptake and the loss of water. So you have a wilted plant because you forgot to water it. It's going to be flaccid, wilted, limp, whatever you want to call it. Flaccid is a cell placed in an environment with a higher solute concentration. So if you think back to the picture, you've got your beaker and you've got your thing in the beaker. Okay, here's a beaker on my desk. Let's say it's full of water. I put this thing of dried crickets. Shockingly, this is a semi-permeable membrane full of dried crickets. My dried crickets are big, so they can't get out. But they're in my beaker of water. What's the water going to do to try and dilute the crickets? Right? It's going to rush into my cricket bag, my bag of crickets. And this is going to make the cell bigger. In this case, I'm going to have a cell is placed in an environment with a higher concentration. So in this case, I have, I don't know, a pink marker. And it's at the outside is a high concentration. The inside is a low concentration. It's going to rush in to my marker. It's going to rush out of my marker into my beaker, and it's going to make it limp. It's going to shrivel up. If that makes sense. I don't know if the beaker actually helped there. So you have a beaker, you have something in it, like a marker. If your marker doesn't is in an environment where the outside has more stuff in it, the water's gonna go out. The cell will lose water and undergo plasmolysis. So the environment has a higher solute concentration than the inside. So that concentration is, so water's gonna rush out to dilute it. This is called plasmolysis, occurs when the protoplast shrinks and things shrivel. Just like this. I should have just used this picture to begin with. So here's my cell, just a normal cell. I'm going to put it in here with a water potential. Here's the water potential. Out here is high. I've got it, it's in a sucrose solution. So I put my cell in a sugar solution because I don't know why, I want it to be sweeter. I want my plant to be sweet. So what's going to happen? It's going to actually lose water. It's going to actually come out here and our plant's going to shrivel up. You could put it in a salt solution. You could do it in whatever you want to do. And it's going to come out here and your plant's going to shrivel up. This is called plasmolysis. We would call it wilting because that's what's going to look like, all going to shrivel up. The next one is going to be a turgid cell. So you're going to see what this looks like in just a second. So now I've put my cell in here and stuff's all going to come in. It's going to rush in and it's going to make my cell all big and happy. Big, fat, and happy. So this is my initial cell, and now my cell's all big, fat, and happy because I'm in pure water. This is why, this is a fish example. Let's say you could buy fish from PetSmart or whatever, and you put your fish in deionized water. Dude, what's going to happen to the cells? It's pure water, right? The water's going to be like, dilute the fish, get in there and dilute the fish. That's why you really shouldn't put your fish in DI water. There should be a little bit of some gunk in there to keep this from happening because your fish will explode. Okay. So there again, there it is bigger, your flaccid cell, and then you're gonna see this water coming out because it's in a sugar solution. Oh, here's a video. I don't know if it'll work. Let's try it. You can actually see the little things moving in there. Okay, let's move on. Turgid and wilting. If a flaccid cell is in a solution with a lower concentration, the cell will gain water and become turgid. Turgor loss is wilting, which can be reversed when the plant is watered. So wilting isn't a permanent condition, usually, unless you wait too long. Then you get wilting. Turgid, wilted. That's sad. This is a sad plant. Aquaporins. These are the transport proteins in the cell membrane that facilitate the passage of water. Good times. So these affect the rate of movement of water. They're just little pieces of the cell that are going to set in there and compete with things. Aquaporins. Okay, I want to talk about bulk flow. Bulk flow is the long distance transport. This is like the tall trees, right? You got to get all the way up there. That's bulk flow. You're sending a bunch of stuff up. Long distance transport. This requires bulk flow. Water and solutes move together through the vessel elements of the xylem. And that's what I want you to know there. Big, fat, bulk flow. Okay. Transpiration drives this process. You know this. This is review. This is all review. You know transpiration. You know xylem and phloem. Plants can move a ton of water, right? They got xylem. This is root hairs account for much of the surface area, so roots are moving it up and it's coming out the other end, right? This is what's happening. You know this. The concentration of minerals is greater in the roots than the soil, so you get active transport. We are skipping this. We are skipping this endodermis business because we didn't talk about apoplastic. I'm not even going to talk about the Casparian strip. This is free information. There's just a little strip here that keeps not just anything from getting in a plant. It's kind of like a little border control, like, hey dude, stop, what are you? What I want to talk about, again, is transpiration. So here we are with transpiration. So skip down to transpiration and xylem sap. You'll see it at the bottom somewhere after Casparian strip. That's where we're starting, okay? That's where we're starting. Bulk flow. This is the big tree. Dude, how are we getting this up there? How are we getting this up? This is review. You know this, right? Xylem sap, it's just the water that's going up in the dead straws. Water and some dissolved minerals, mostly water. This is transported from the roots up to the top of the trees. We're doing this down here. We've got the little root cells. We got water uptake from the soil. So water's going in to dilute the root, right? Water's going in, and then it's just coming right up to the top. In through here, we have cohesion and adhesion. These are properties of water. You learned it in chemistry. The awesome thing about water is it adheres to each other, right? This is what makes water fountains possible. The water holds onto each other, so it makes the little fountain, right? Then you have adhesion by hydrogen bonding. So both of those things are happening by hydrogen bonding. It's causing them to stick to each other, and it's causing them to stick to the sides of the little dead straws. So it's sticking to the sides of the dead straws, they're sticking to each other, and they're pulling each other up through transpiration. Again, it's all about transpiration. It's required as water travels up the roots. Okay, this is cool. I love this. This is fun. There's a little bit of root pressure, so let's go back. Is replaced as water travels up from the roots. The roots are like, come on, go, go. It's like a poking. Go, dude, go, come on. This is not huge. This is not what's driving the process. Do not be confused. Transpiration is driving the process. But roots are like, dude, go, come on, go. Kind of poking them, go, go. And this causes this process here, which is super cool. It's called guttation. Guttation is like the roots, and so this is a little bit of xylem at the tip of this roots. This is not dew, this is guttation. So is it pushed by the roots or pulled from the leaves? One of them is, it's kind of pushed a little bit from the roots. At night, the root cells are still pumping stuff, but nobody's transpiring because nobody's photosynthesized. So the plant's like, settle down. Down there, roots, settle down. And the roots are like, go, come on, go. And you get, boom, you get that little tiny bit. So it's not spewing out. You don't walk past pants and they're like shooting you in the eye with xylem, right? No, that's not happening. Just sometimes in the morning you see these tiny droplets. It's not dew, it's guttation. So you do get a slight root pressure. Root pressure is sometimes called guttation, and it happens right on the edges. And there's some examples. Oh, is it dew? No. This is guttation. This is dew. If it's like a bunch, it's dew. That's the condensation from the air at night. This root pressure is relatively weak and is a minor mechanism. Again, transpiration is driving this process, not root pressure. Cohesion tension. Let's talk about this. Cohesion versus adhesion. Both are hydrogen bonding. Cohesion pulls the water from roots and shoots. So does adhesion. So there's some cohesion and some adhesion. Xylem sap is under negative pressure because it's ready. It's ready. Just push it up. It's ready to go. Why? I really want to do here. Hold on. Why is it not doing that? Okay, I just want to make you a little slide here of this because it's in your notes and I want to make sure you have it. So let's start with, we start with adhesion. Adhesion. What is adhesion? Adhesion is between a water molecule and other polar substances. So this is the water molecule attaching through hydrogen bonding to the side of the xylem. This is it's adhering to the side of the wall of the xylem, which is allowing it to crawl up the dead straw. So what is cohesion? Cohesion is between the same water molecules. So this is water holding on to water. This is water molecule, and they're holding dry, like, it's like Barrel of Monkeys. Have you ever played that where you hook the monkeys? All the monkeys are hooking on and pulling up the dead straw until it's transpired. So the monkeys are holding on to each other, and the monkeys are kind of holding on to the side. Your water molecules are like a barrel of monkeys, and they're pulling themselves up through cohesion, and some of the monkeys are sticking to the side through adhesion, and so they're able to crawl up through transpiration. So that is adhesion and cohesion, properties of water. What else do I want to say about that? There's a little bit of pull on that, and what we saw earlier is if you break that off, you are in trouble. That's what happened with our That's why you had to submerge the plant under the water, because if you get an air bubble in there, then it ruins your adhesion and your cohesion, and it messes up your bulk flow. So that's all I'm going to say about that, and I'm going to make one more video, hopefully.